Hi, I'm Jessica Wegman. I'm a master's student in the experimental psychology program here at RIT, and I work in Dr. DeLong's CCP lab. In this project, we examine visual object categorization in goldfish. What is categorization? It's the process where objects that are not the same are mentally grouped together based on their defining features. For example, chairs come in all different variations of shape and size. However, when you see a chair, you're able to identify it as such, even though it probably looks different from the first chair you ever saw. You don't need someone to teach you each time that you encounter a new type of chair that it is a chair. You quickly categorize it, and then you're able to properly use it by sitting in it. Categorization is a useful ability that many non-human animals have been found to possess, such as monkeys, pigeons, rats, and baboons. Categorization could potentially aid goldfish in a variety of different activities that help with their survival, such as finding food, mates, and identifying predators. Categorization has been studied in one other bony fish species, cichlids. In a previous study, cichlids were trained in a two alternative forced choice task to discriminate between a fish and a snail in black and white 2D stimuli. They were then presented with an array of different fish and snail drawings and later pictures of different kinds of fish and snails. Some examples can be seen here to the right. Cichlids were able to categorize these stimuli into two categories of fish and snail with both line drawings and actual photographs of these different animals. In the present study, we wanted to examine categorization abilities in goldfish. Specifically, we wanted to understand the type of features that goldfish use to categorize turtle and frog 3D stimuli. We started first with two training pairs, a turtle and a frog, that you see to the right. Then we presented the fish with an array of many different frogs and turtles that varied in color and shape. So it was thought that if the goldfish were categorizing based on color, they'd choose stimuli like these ones, where the colors match the training stimuli, but the shape of them do not. If the fish were instead categorizing based on shape, then we'd expect the fish to choose stimuli like these, where the shape matched the training stimuli, but the color did not. 7 fish participated in the current study. They were trained and tested 5 days per week. Before the beginning of training, the fish were randomly assigned to either the frog S+ or the turtle as their S+. During training, the fish had to reach a criterion of 75% overall accuracy in order to move on to the test. Once the fish progressed to testing, they were presented with an array of 12 different turtles and 12 different frogs that varied in shape and color. There were 6 trials per session and 24 total sessions in the entire testing block. All 7 fish completed one full block of testing. To the right is another picture of a fish during a testing session while they are choosing one of the 2 stimuli presented. In the study, a 2 alternative forced choice task was used. For both training and testing, fish were first moved from their home tanks into individual testing tanks. During a session, the fish first had to face the back of the wall and before a trial could begin. At this time, the stimulus board was inserted into the front of the tank. The fish were then able to swim up to the stimulus board and would tap one of the stimuli using their mouth to make a choice, which you can see a fish doing so in the top picture. If they were correct, then they would receive a food reward from a syringe, which you can see in the bottom picture. If they were wrong, the stimulus board was removed and the fish would move on to the next trial without a food reward. There was an inter-trial interval of about 15 to 30 seconds between trials, and the fish were never food deprived, no matter their performance. Once the fish were trained with this one pair of training stimuli, they moved on to the test. The test stimuli included this array of novel turtles and frogs that varied in color, shape, and size from the training stimuli. The figure on the right shows the full array of test stimuli, which includes 12 total frogs and 12 turtles. These test stimuli could be characterized into 2 ways, shape and color, with the first character being shape, There were more rounded frogs, which matched the training frog shape, in addition to chunky, skinny, and curved back frogs, which did not match the training frog shape. You can see from these representative samples the types of stimuli here, but there were multiple stimuli within each group. These weren't just the only ones presented. Then the turtles were the other test stimuli, and there were some high round shell turtles, which was the same shape as the training turtle, in addition to turtles with flat shells, sea turtles with flat shells and ridges, high spiky shells, and high oval shells with different shapes than the original training turtle. Again, these are just some representatives of the stimuli within these groups, but there are multiple turtles within each characteristic. The second way we characterized the test stimuli was by color. For the frogs, there were some that had colors that matched the training frog, which is labeled as F1 here. These stimuli were yellow and then also red or green within the yellow. There were also green frogs whose color matched the training turtle, which is labeled as T1 here. And finally, there were brown, black, blue, and pink frogs that did not match the original training stimuli based on color at all. We had the same categories for the turtles. So there were some green turtles that matched the original training turtle, T1, in color, some yellow and orange turtles that matched the color of the training frog, F1, and some brown turtles that did not match either the training frog nor the training turtle based on color. Getting into the results, the graph on the left shows testing performance accuracy based on the assigned S+ for each individual fish. Here we can see that performance was significantly better for fish that were randomly assigned to the turtle S+ than for fish that were randomly assigned to the frog S+. The graph on the right shows testing performance accuracy for the assigned S+ as an average of each individual fish within that group. There were no significant differences between fish with the frog S+. For fish with the turtle S+, Kanaloa and Pele performed significantly better than Maui did. It is common to find individual differences between fish subjects based on previous research that is similar to this study, so some of these differences were expected. This graph shows how the fish were randomly assigned to the frog as their training S+ versus those that were assigned to the turtle as their training S+ performed as a function of the presented test S+ color. The items that I've marked with blue represent the group of test stimuli that match the training S+ color. The items I've marked with green represent the group of test stimuli that match the training S-. And finally, the items I've marked with red represent the group of test stimuli that do not match the training S+ nor S-. For both groups of fish, if the test S+ color matched the training S+ color, they performed well. That is shown here by the blue circles. If the test S+ color matched their training S- color, the fish performed poorly. This is shown here by the green circles. Finally, if the color of the test S+ did not match the training S+ or S-, and it was a completely different color, then the fish performed poorly if they were assigned to the frog S+, but they performed well if they were randomly assigned to the turtle S+. This shows us that the fish assigned to the turtle S+ used shape in addition to color to categorize stimuli, but the fish assigned to the frog S+ only used color. This graph is similar to the previous one, but instead of examining test S+ color, we examined the test S- color. So here we're looking at how the fish who were randomly assigned to the frog S+ versus the turtle S+ performed as a function of the S- test stimulus color. Again, the items that I've marked with blue represent the group of test stimuli that match the training S+ color, and the items marked with green represent the group of test stimuli that match the training S- color. Finally, the items I've marked with red represent the group of test stimuli that do not match the training S+ nor S- colors. We found that there was only one condition where the fish assigned to the frog S plus performed well, which was when the testing S minus color did not match the training stimuli at all. This is shown by the red circle for the frog S plus group. For the turtle S plus group, it was the exact opposite. The fish assigned to the turtle S plus performed well when the presented test S minus color matched the training test or the training S plus color, as well as when the testing S minus color matched the training S minus color. In both cases, this shows that fish could categorize on the basis of shape, either when there was no color match to the frog S plus group or when the color was in conflict with their choice for the turtle S plus group. The main takeaways from this study, which is the first to investigate categorization of 3D objects in goldfish, is that there were individual differences in performance among fish. Again, this finding is consistent with similar previous research. The second point that we found is that goldfish were able to categorize 3D stimuli based on both color and global shape, which was the turtle and frog groups. Finally, we found no difference in performance within the various shape groupings, such as skinny frogs and round frogs, or the flat-shelled turtles and the spiky-shelled turtles. And based on these findings, future research should investigate the ability of goldfish to categorize based on shape without any color present. I'd like to thank the funding agencies that made this research possible. If you have any questions, please email either me, Jessica Wegman, or Dr. Carolyn DeLong. Our emails are listed on screen. Also, if you're interested in this research, you can visit the RIT CCP Lab website to read more. Thank you for watching.